Okay, we are on to the back of the Genetics Problem 1 worksheet. Genetics Problems 1 worksheet. Let's go ahead and look at number 4 here. The gene for the length of whiskers has 2 alleles. Again, I kind of, as a general rule, kind of underline those biology words. Things like dominant allele W, codes for long whiskers. So I'm just going to take long whiskers here. I'm just going to put a note out here since it's hard to kind of draw an arrow to that. I'm going to put DOM for dominant. Okay? And the recessive allele, little w, codes for short whiskers. I'm going to circle short whiskers here. I'll just point over to recessive. Let me know this is the recessive allele. What percentage, again, we're looking for a percentage here, of offspring would be expected to have short whiskers? So we're looking for short whiskers from the cross of 2 long-whiskered seals. One that is homozygous dominant. This is parent 1. I'm going to put P1 down at the bottom. And one that is heterozygous, parent 2. That's going to help us with our step 2. So, step 1, as always, you guys know, is make a key. Make a key. Okay, dominant allele, W, codes for long whiskers. Okay, a recessive allele, little w, and I'm going to make it extremely small because these are easy letters to confuse the uppercase and lowercase. So if that's the case, try to exaggerate how small the w is. That's what I do anyway. Short whiskers. Little w equals short whiskers. Okay, we have a key. 2. Parent genotypes. Okay. Parent 1 is homozygous dominant. Homo, remember homo means same, right? Same. Hetero means different. Different. Okay. Homozygous dominant parent, that's going to be big 8, or oh, I put H. I changed it without thinking. Big W, long whiskers. Homozygous dominant. So we have a big W, crossed with a heterozygous individual, right? So the second parent is heterozygous, meaning different. Big W, little w. Okay, we have our parent genotype. Step 3, cross them. Okay. Let's go ahead and put big W up top. And I'll put big W, really little w, over here on the bottom here. And then this part you guys should know pretty well by now. Had enough practice probably. Big W, little w, big W, little w. Okay, it asks you for, what is it asking for here? The percentage of offspring that would be expected to have short whiskers. So short whiskers is a recessive trait. So the only way you can get short whiskers is little w, little w. Okay, how many of these little w's do you have in here? Well, I'm glancing at this and I don't see any of those, which means it's not a trick question. This is just 0%. 0% short whiskers. Okay, that's it for that one. Let's move on. All right, this is kind of a long one. This is our first dihybrid cross here, but the process is mostly the same with the exception of a little part in the middle that we'll talk about. I have another step we're going to kind of add to this one, but number 1 stays the same. But let's read first. The green color allele is dominant over yellow, so I'm going to circle green. Green color is dominant, right, over yellow color, which is recessive, right? Recessive for seed color. And tall is the dominant allele in plant height. So for plant height, tall is dominant. So I guess short we can think of as recessive. It doesn't specifically tell that to us, but I think we can assume that, right? Short is the recessive. I'm just going to write that out to the side so I remember. Parents heterozygous for both traits are cross-pollinated. Determine the frequency for the 4 different phenotypes of the offspring. So So frequency is just a fancy way of saying percentage without the multiplying times 100 part. So step one is the same. First thing we do is make a key. Make a key. Okay. Well, we know green color is dominant to yellow, so I'm just going to put a big G. That's going to give me green. Little g is going to give me yellow. All right. We know T is the dominant allele for height, and we know that tall is the dominant allele. So I'm going to put T tall, and little t will make that equal to short. Okay, now we have a key. Second step. What are the parent genotypes? I'm just going to put GTs again for genotypes. So leave myself some room here. Parent genotypes. Well, it says parents heterozygous for both traits. All right, so if a parent is heterozygous for both traits, let's go ahead and do parent 1 here. Big G, big T, little t. That's parent 1. The other parent is also heterozygous, meaning different for both traits. Big G, big T, little t. Remember, hetero, different. 3. All right, the third thing, and I'm going to change this step for simply when we're doing dihybrids, we kind of have to find out our gametes. All right, we have to find out what gametes each parent could give up. And I'll just label this gametes. And I'll put in parentheses foil because we're going to use the foil method to figure out every possibility of G and T, remembering that each parent is going to give up one of the G alleles and one of the T alleles for each gamete. So let's do parent 1 over here, and let's foil this. I'm just going to put down here F for first, O for outside, I for inside, L for last. Okay, let me scroll down a little bit, see if I can find myself some room over here. So first parent is big G, little g, and I'm going to spread out this genotype a little bit, big T, little t. And I'm going to put them in parentheses. You don't have to do this, but if this helps you from a math perspective, you remember, when you factor polynomials or multiply them back in, you kind of come up with this, it's kind of similar to that, using that FOIL method when you're multiplying into parentheses from something else that's in parentheses. First thing you got to do is I look at the first one of each of these statements in parentheses. So first one here is G, right? I'm just going to be 1. And the first one on the other side is 2. First one in these parentheses and the first one in these parentheses. So my first one is going to be big G, big T. Okay, let me erase here. All right. Now we need to find the outside. When I talk about outside, I'm talking about what's on the furthest to the outside of everything. Well, the big G is furthest from the big T. That's outside. These are on the outside of the entire expression here. So I'm going to put big G, little t. Okay, let me erase those. Okay, inside, let's look at little g, big T. Those are going to be the inside ones. Which ones are on the inside here? Little g, big T. All right. And then finally, let's look at last, which is going to be the last in each set of parentheses over here. That's going to be little g, little t. All right, we now have unique gametes for one parent. Now, you'll notice both of these parents have the exact same genotype. So really, this other parent's going to give up the exact same gametes. Big G, little t, big G, big T, little g, big T, and little g, little t. Okay, now and only now can you do step 4, which is cross them. Okay, so all you got to do is take one of these parents. They both have this, can give up the exact same, make the exact same gametes. It doesn't matter which one, but big G, big T, I'm going to do this one here. And then big G, little t. I'll put a little dot down there so you can follow along. Little g, big T. Little g, big T. And then little g, little t. G, little t. Okay, same thing on the other side. Big T. Big G, little t. Little g, big T, and little g, little t. Okay, I'm going to go ahead and do one of these, and then I'm going to let you guys kind of complete this on your own. Let's see. Big G, big G, big T, big T. Now, one thing to note about dihybrids that I've seen the mistake that people make, if your G's are first, always keep your G's first in your genotype, right? Within each of these letters, you want to keep your dominant in front, right? You always want to keep your dominant allele in front of the other one. So I'll give an example here. Let me find a good spot here. Let's go to this. I'm going to go to this square down here. So this one I have big G, little t, crossed with little g, big T. So I'm going to put big G, little g, right? I'm putting my G's first, and I'm going to keep them that way. But I always put the dominant G in front of the recessive G. Now I also have big T, little t over here. I'm going to keep my T's second, but I'm always, within my T's, going to keep my dominant first and my recessive second. Okay, I'm going to pause the recording and let you guys finish up. You guys can pause the video if you like, because I'm fixing to complete this whole thing, and then we will come back and do this other part. So go ahead and complete this, and then restart it when you're finished. Okay, so we're back here. I've gone ahead and finished this whole thing out. So we want to know what percentage of these, and this is somewhat familiar. We're looking, these are phenotypes. Tall plant green seeds. Let's see how many of those we have. And there's multiple ways to do this. I'm going to go ahead and just select green here. And I'm going to go by seeds first. We know green is dominant, right? Green is dominant over here. So if I have a big G, it's anywhere. Even if it's just one, it's always going to be green. So I'm just going to put a green circle on all of these that are green. Here's a green, here's a green, here's a green, here's a green, here's more greens. Again, all it has to have is a big G. All right, so a lot of greens here. Okay, now let's look at the other allele here. Just let's look at yellow. I'm going to do this right now since we're talking about seed color. I'm going to go ahead and label in yellow all of the yellow plants. Now remember, yellow is represented by a little g allele, meaning that the only way you'll get yellow plants is if you have two little g's, which there's only 4 boxes of those. Let me label them with yellow down here. Okay, let's look at the second trait here, tall and short, and let me see if I can find some more colors. We'll put tall. We're going to look for red. So now we're looking at the second trait. Anywhere you have a big T, that's a tall plant. So I'm going to put a red circle wherever I have a big T. All right, red, 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 red. This one has a big T. This one also has a big T. Big T. Down here. And big T's all across here. All right, let's look at our short plants. Again, short plants can only have a little t, right? So we're gonna use a different color for that. Let's use this cyan, this blue right here. Little blue is gonna be short. So little t, that's gonna be short. Little t again. Little t, little t, little t, little t. Okay, we've got the phenotypes for each of these. The first one we want to look for, and I'm going to use my color coding here, is tall plants, which if you remember, tall is dominant, so we're looking for the red, and we're also looking for green seeds. So all you have to do is count those up. So let's see here. We're looking for things that have both a blue and a green circle. And I'm just going to switch back to black here and kind of mark them off right here. 2, 3, 4, 5, 6, 7, 8, 9. Okay, I've got a total of 9. You may want to check and see how many you have. But there's 9 total. If you didn't get 9, something went wrong. Make sure to figure out where you went wrong. But I got 9 out of a total of how many squares? 16. A total of 9 out of 16 are tall and green. Now, if you put that in your calculator, let's see here, 9 divided by 16, you're going to get 0.5625, which is the way we typically represent frequency. So I'm going to put 0.5625. As long as you get 9 over 16, 0.5625, that's fine. All right, let's move on to the next one. Next thing we need to find is tall plant yellow seeds. So, tall plant, we're looking for red circles. Yellow seeds, we're looking for yellow circles. Okay, so anything that is red and yellow is now what we're going to want to mark. Let's look for red and yellow. Okay, here we go. Red and yellow down here in the corner. I see 1, 2, and 3. So, 3 out of 16, or let's put it in our calculator here. 3 divided by 16, we got 0.1875. 0.1875. Okay, same thing. Let's move on to the next one. If you've got it from here, maybe finish it up, and then we'll come back to it. The next one is short plant. So we're looking for the cyan, because this is short plant, and green seeds. Select the green over here, underline it. So we're looking for cyan, or blue, and green. Okay, let me switch back to black. So blue and green, I got 1 here, 1 here, and 1 here. So 3 out of 16. And we already calculated this one, so you can just kind of copy it down. 0.1875. And finally, we got to find our short plant, right? Cyan and yellow seeds. Underline it in yellow here. And of course, you probably notice that there's only one of these that does not have a dot by it, and it's this bottom right one, the rarest phenotype here. Short plant yellow seeds, 1 out of 16. Of course, if you put that in your calculator, you're going to get 0.0625. 0.0625. Okay, that is how you do a dihybrid Punnett square. Now below, I'm going to teach you a little bit of an easier way to do it. This is a bit of a shortcut, but we're going to do the same thing down here. So keep in mind our key, and I'll rewrite it down here a little bit. Okay, shortcut way of solving the same dihybrid cross. Because of Mendel's second law of independent assortment, you can work the color gene and the height gene separately. So we're going to set up 2 separate monohybrid crosses. So we're going to look at the first gene, which is color, right? So the G's. So we're going to cross the first parent, big G little g, with big G little g. So this is parent number 1. Just going to look at these alleles separately. Big G little g. Okay. Let's set that one up. Let's go to the second gene we need to talk about, and that's the height gene right here. Big T little t crosses big T little t. Let's cross that one as well. Big T little t and big T little t. Okay, let's do our crosses here, as I'm sure you're used to by now. Big G, big G, big G, little g, little g, and little g, little g. Do the same over here. Big T, big T. Big T, little t, big T, little t, and little t, little t. Okay, now we can use the laws of probability to calculate our frequencies, which we're going to use later on in one of these worksheets. But let's look at height first. We're looking for tall plants, green seeds. Tall is, remember, is our dominant allele. Green seeds is also our dominant allele for color. So, for the first one, this is our seed color. I'll put seed color over here, and this is our height. Okay, so for height, we're looking, or for seed color first, we're looking for green seeds. Okay, 1, 2, 3. 3 out of 4 are green in color. Okay, let's look at height. How many of them are tall or show the dominant phenotype? 1, 2, 3. 3 out of 4 as well. So let's just put 3 out of 4 here. Now, if you multiply these together using the laws of probability, you're going to get 9 over 16, which should look somewhat familiar. If I scroll back up here, look up here, didn't we get 9 out of 16 for tall plant green seeds? We did down here as well, right? 9 out of 16. So we can do the same for all of these. Tall plant, again, dominant. Yellow seeds is, I'm going to put an R for recessive for the seed color. So only 1 out of 4 for color. So I'm going to put 1 out of 4. And of course, tall plant, 1, 2, 3 out of 4, 3 out of 4. Multiply them together, and you get 3 out of 16. Okay, short plant, green seeds. Short plant is recessive, green seeds is dominant, right? So, for height, short plants, it's the recessive, 1 out of 4. Color, green seeds is 3 out of 4. Multiply those together, and you get 3 out of 16. And then finally, short plant, yellow seeds, both of these are the recessive. So let's look at the short plant. Little t, there's 1 out of 4, only 1 out of 4 of those. Let's look at little g over here on the other side. Only 1 out of 4 of those. Multiply those together, and you get 1 out of 16. This is one of the most important ratios, so you might want to put a star by this or something to know. 9 to 3 to 3 to 1. Of course, the 9 represents the dominant for the first gene, dominant for the second gene. The 1 represents recessive for both, right? And the 3s represent dominant for one, recessive for the other. And the 3 represents the recessive for the other one and dominant for the other one. So, an important ratio to know, very, very important. Write important up here. So make sure you know that, and make sure you know how to solve a dihybrid Punnett square. Okay, that is it for worksheet 1.